All right, in the first video for this chapter, we defined the glomerular filtration rate, the GFR rate, as the amount of filtrate that's produced by the kidneys each minute. And so now we need to look at what is regulating this GFR. And so really there's three types of regulations. Regulation for GFR, it's either automatically controlled, and we'll see that first through a couple different mechanisms. Hormonally controlled, and so we'll go back through a few more hormones, most of which we've already seen before, and then also through the nervous system, through neural control. And so let's look at the automatic type control here. To begin with, we need to look at some vocabulary words, and you notice that these are kind of some strange words we haven't really seen before: juxtaglomerular apparatus, juxtaglomerular cells, and macula densa. And so let's just kind of look at the diagram for a minute, kind of orient ourselves again. You have the nephron listed here, kind of shown here, and then they enlarge it and they kind of show you a big picture here of the renal corpuscle. And of course that has the glomerular capsule on the outside, or the Bowman's capsule on the outside, and all the glomerular capillaries on the inside. And then you have an afferent arteriole coming in, bringing blood in, and to be filtered, and the efferent arteriole leaving it. And then over here to the side, you have a portion of the distal tubule, and you can kind of see from the small view here that distal tubule is coming up from the loop of Henle, and then it kind of curves in really close to this zone. And now this area here is what's called the juxtaglomerular apparatus. It is this zone here where all these things kind of come together. The distal tubule is here, the efferent arteriole and the afferent arteriole all kind of come together in this zone called the juxtaglomerular apparatus. Now, really that includes two parts. There are juxtaglomerular cells, and there's something called the macula densa. And so the juxtaglomerular cells are essentially smooth muscle cells. And so in this particular diagram, they're kind of labeled here as blue or purple, kind of purplish color here, and they essentially wrap around and create almost like a little rubber band around the efferent and the afferent arterioles. And so that means that because they are smooth muscle cells, they can constrict and dilate. And so whenever they contract, they constrict, and that's going to decrease the amount of blood flow in and out of the renal corpuscle. And then they can also relax and dilate, and that increases how much blood flow is going in and out. And so we need to talk about that in a form of autoregulation. The macula densa cells, we'll see this in a minute as well. These are cells, specialized cells that are located in the distal tubule, just right around this area. And these have the ability to detect a change in flow of filtrate, and that sends, that they're right up against the juxtaglomerular cells, and so that can send a signal here to the afferent or the efferent arteriole to either constrict or dilate. So all of this type of system here is trying to regulate the flow or the production of filtrate by controlling how much blood is coming in, how much blood flow is going into the nephron, renal corpuscle here, versus how much is coming out. So this is called autoregulation. Like I said, there's three different mechanisms that we'll look at. The first one is autoregulation. And under autoregulation, you have two main types here. One is called the myogenic response. So first of all, look at that word myogenic. Myo, of course, is your prefix that refers to muscle. And so this is generated by muscle. This is essentially those smooth muscle cells that I just mentioned for the juxtaglomerular apparatus cells. And it involves a stretch reflex. And so just like many other smooth muscles, whenever something causes them to stretch, it causes those smooth muscle cells to contract, and contraction causes constriction of those vessels. So you have to think, well, what would cause these smooth muscle cells in this area to stretch? And usually that would be a higher amount of blood volume, usually created by creating a higher blood pressure. So higher blood pressure and blood volume go together. And so if your blood pressure is going up a little bit, to try to regulate your GFR, to try to regulate how much filtrate is being produced per minute, then your afferent arteriole would constrict. So let's go back and take a look at this. And so let's say that blood pressure is going up. If this didn't happen, you would have added blood flow coming in, and it would kind of overload this nephron. You would have more blood flow coming in than it would be able to handle. And so to kind of minimize the amount of blood flow coming in, if your blood pressure is going up, then the afferent arteriole will constrict, and it will, decrease how much blood flow is going in. And of course, the reverse is true. As blood pressure, goes back down, or if it goes down a little too low, then your afferent arteriole can dilate, and that just tries to help fluctuations, try to minimize the fluctuations in the GFR. Now, the second type of autoregulation involves the macula densa cells that we talked about for the distal, that's inside of the distal tube. And this is what's called tubulo-glomerular feedback, because it has something to do with the glomerulus and also has something to do with the tubes, in this case the distal tube. What's happening now is that it's trying to detect how much filtrate is coming through the distal tubule. And so again, these cells are located here, and you can see this diagram. It's a little different than the first one I showed you, but here's your distal tubule. It's right up against the juxtaglomerular cells here. Here's your afferent arteriole coming in, bringing blood into the renal corpuscle for filtration. And so let's say you have extra flow rate coming through. Maybe your GFR rate is a little too high. Well, you need to minimize that. You need to balance it back out, create a homeostasis type environment. And so maybe the filtrate rate is coming up too high, so that means that this distal tubule would stretch a little bit. That increased flow rate through here stretches, and it transfers that signal because it's right up against those juxtaglomerular cells, and so it just by its touching into those cells, contacting those cells, it's causing those cells to receive that information, and they begin to constrict. And so if you have too much filtrate coming through here, that's another signal, a way to autoregulate and to decrease how much blood flow is actually going into the renal corpuscle. Now, another way that GFR can be regulated or is regulated is through hormones. And so again, you have, it still does involve the juxtaglomerular cells, so this is still here. Let me give you a scenario where hormones would try to help balance out your GFR. This is through a system called the renin angiotensin aldosterone system or pathway. Sometimes it's shortened RAS, and it involves several chemicals, and I'll show you kind of a flow chart of different hormones / enzymes that are used to try to balance out your GFR rate. And so let's say, for example, you have low blood volume or low blood pressure, and your juxtaglomerular cells that we just talked about detect that there's a reduced amount of stretch in the afferent arteriole. So that's where we're at. Here's our renal corpuscle, afferent arteriole is here. Maybe there's a lower blood volume coming in. And so the question is, how can you fix this? If the lower, you have lower blood pressure, maybe it's due to lower blood volume. What types of things can you do to remedy this? And so one of the things that can happen is a chemical can be secreted called renin. Now, in this little flow chart, it kind of shows you the pathway here. We've seen some of this before, so let's kind of go through some of these terms again. Renin is produced by the kidneys, and it's produced whenever there is low blood volume going into the renal corpuscle. It is technically a hormone, but it works like an enzyme. So a hormone that's produced, it's produced by the organ and goes into the bloodstream, and it has a Dyson effect and all that. But it really works as an enzyme by speeding up a chemical reaction. That chemical reaction is listed here for you. So there's a chemical from the liver called angiotensinogen. That chemical, in the presence of renin, angiotensinogen gets secreted in the blood. And so if renin is secreted, it's also in the bloodstream, and that will cause angiotensinogen to be converted to a different chemical called angiotensin 1. Well, angiotensin 1 will be converted into angiotensin 2 by a different chemical called angiotensin converting enzyme. This is also called ACE, and we've actually mentioned that before. If you go back and look at the respiratory system, we mentioned that one of the functions of the pneumocytes, the lungs, is to produce this chemical called ACE. Angiotensin 2 has several effects. One is that and I've listed several of these out here for you. One is that it increases your thirst receptors. So your hypothalamus is kind of in charge of some of this, and so it makes you feel thirsty, and so that helps you to increase your body fluid balance, your body fluids, and that will hopefully help to increase your blood pressure. It also triggers salt appetite, and so now you have a feeling that you want something to eat salty. Increase the salt intake, and that should probably follow by, or that will probably lead to an increase in fluid intake. It also increases the ADH secretion, antidiuretic hormone, so you're retaining the fluids that you have already because you don't want to waste any more fluids because you're already low on body fluids and blood pressure. It also increases other hormones we've looked at, aldosterone. Aldosterone helps to retain sodium, and so if you retain sodium, you also retain body fluids. That's based on osmosis. And then it also causes vasoconstriction in certain vessels, because vasoconstriction, if you decrease the diameter of a vessel, that also increases blood pressure. So all of these things here will lead to an increase in blood pressure. By the way, it's the angiotensin 2 that triggers the aldosterone to be released. Remember, we've talked about this a while back from the adrenal cortex. And so that's one of those corticosteroid hormones that we talked about that comes from that cortex area of the adrenal gland. There's a figure 24.14 in your textbook that does a pretty good job of showing you each of these chemicals and things and where they're produced from. You see the blood vessel there, and the liver's producing the angiotensinogen. The kidneys are producing renin. If you have a decrease in the GFR and you've got blood pressure going down, then the kidneys are going to secrete the renin here. That's converting to angiotensin 1. The ACE is coming from the lungs, and that converts it to angiotensin 2. And then you can kind of see all the different effects we just talked about listed there for you. And then at the end, it should lead to blood pressure increasing, and it should lead to your GFR returning back to the normal, the normal balance. One other thing about aldosterone we mentioned before is it helps to retain sodium, but we never really went to a kind of a molecular cellular look at what it's really doing. And so one of the ways that aldosterone helps to absorb or helps your body to retain more sodium is by increasing the number of transport proteins that you have. And so here you're seeing a tubule cell, like one of the like the distal tubule type cell and the proximal tubules. And so the idea is to aldosterone, that hormone, goes inside of these tubule cells. This is actually inside of the cell, not inside the tube. So down here where it says lumen, that would be inside of the actual tube. And so that's where the filtrate would be. But you would have receptors in the membrane of this tube that this aldosterone hormone would bind to, and that triggers the production of more sodium transports. And so by having more sodium transports, that means you can transport more sodium back into, eventually, first of all, it has to go into the interstitial fluid that's out in the tissues, and eventually that'll be transported into the bloodstream because there will be a blood vessel, capillaries, or something out here, and that extra sodium now gets transported back to the bloodstream. Well, the main thing here is that once sodium goes in that direction, so here's this sodium-potassium pump here, it's pumping sodium out. Notice it does require energy to be used, so it's a true active type transport, and so it's pumping the sodium out, and then water will want to follow that after the sodium goes out, and that's because of osmosis. There's also a negative feedback loop, so it's the higher levels of sodium that basically stop the production of the renin. So once you go back here, when we said renin is produced here, well, what turns off the production of renin is by having elevated levels of the sodium. So once sodium goes up, then that backfeeds as a negative feedback loop and causes the renin to slow down production. Another hormone that we've talked about before is ANP, atrial natriuretic peptide. And so I just listed some of those notes that we've looked at before here. It is produced by the right atrium. We've talked about that whenever the blood volume increases the cells and increases the stretching of those cells inside the right atrium. Basically, you can think of ANP as the opposite of aldosterone because it's blocking sodium reabsorption, and it also inhibits ADA production. And this allows for, if you block the sodium reabsorption, and so that basically allows you to get rid of more fluid, and so it increases the volume of urine that's produced. And so whenever you lower that, then you lower blood pressure. And so ANP is also used to help balance out your GFR rate as well. Now the third mechanism here that helps to regulate your GFR is your nervous system. And so you've seen these things before, sympathetic versus the parasympathetic nervous system. Sympathetic nervous system generally speeds things up a little bit. For the most part, it does have it does constrict some of the arteries. Many of the arteries will constrict based on sympathetic nervous system stimulation. But remember, it does also dilate some of the vessels as well, because sympathetic is all about your fight or flight scenario, and so of course you want those vessels going to your cardiac cardiac muscle and to your skeletal muscles and things. You need those vessels to dilate, and so your sympathetic nervous system stimulation will cause those to dilate to get more blood flow to those critical areas. But at the same time, because they have different receptors, they're going to cause other arteries and vessels, especially the smaller ones, to actually constrict, and that decreases blood flow out to certain places such as the kidneys. So under sympathetic stimulation, it actually decreases how much blood is going to the kidneys. That's your renal blood flow rate, and that decreases how much filtrate is formed. Now, your book suggests that under certain conditions, under certain traumatic type events, your sympathetic stimulation could actually decrease your filtrate rate down from around 125 mils per minute. That's your normal GFR rate, just down to a few milliliters per minute. So it really dramatically drops your GFR rate in a very short amount of time. And of course, after the crisis is over, after the traumatic experience is over, then this should return back to your normal GFR rate. All right, so that is filtration. We spent most of our time talking about filtration, but remember there are 3 steps to excretion, one of the main functions for the kidneys. 3 steps there. The first one was filtration. We'll just say a few things about tubular reabsorption for step number 2. Reabsorption is what it sounds like. You have to reabsorb the water and the solutes, and about 99% of the overall filtrate is reabsorbed. It's reabsorbed by these processes listed here, and hopefully you already are familiar with many of these. We'll kind of mention a few things about each one. Diffusion and facilitated diffusion. Make sure you realize that diffusion is a type of passive transport, which means it doesn't require any energy. It's always going from a molecule, is always going from a high concentration to a low concentration. Sometimes you see the word facilitated diffusion. That just means that it needs help. And usually that help comes from some kind of protein channel. The molecule perhaps is too large to go through the membrane on its own, or maybe it has a charge associated with it, or maybe it's water soluble and it doesn't go through the membrane on its own very easily. And so, it needs help through some kind of a channel, an opening through the membrane. So that's facilitated diffusion. Active transport, I've actually mentioned that briefly a little bit ago when we looked at that sodium-potassium pump. But it does require energy, and what active transport is doing is transporting a molecule against its concentration gradient, so it's going from a low concentration originally to a high concentration. Symport and antiports, this is when you are transporting a molecule. Symport means the same direction, and so this is 2 molecules, at least 2 molecules or 2 ions going in the same direction across a membrane. Antiport, that's also 2 molecules, but they're going in opposite directions. For example, the sodium-potassium pump most of the time is an antiport. You're pumping sodium out and potassium is going in. And then osmosis. Some students get osmosis mixed up with diffusion. Osmosis is a specific example of diffusion. It's only applied to water. So if you're talking about water moving across a membrane, then you're talking about osmosis. It is still a passive type transfer, because osmosis doesn't require energy to take place. Now, reabsorption begins in the proximal convoluted tubule. Remember, filtration is all going to be inside of the nephron, inside of the glomerulus and the renal corpuscle and all that, and then you move that filtrate into the proximal tubule, where about 65% of the filtrate is reduced, and most of that is because of osmosis. And so in this picture, you kind of see the cells here, the proximal tubule, and then over close to it, you would have a capillary. And so in this case, a lot of the water from the filtrate, on this side would be the filtrate, a lot of the water from the filtrate actually moves through the cells and is transported out into the interstitial fluid of the kidneys, and then eventually over to the, or interstitial fluid, yeah, over of the kidneys, and eventually to the capillaries in the kidneys. Notice also that the water has a channel as well, because aqueous solutions, water-based solutions, don't easily go through the lipid bilayer of the cells, and so they have a channel of their own. They're called an aquaporin. Those aquaporins are protein channels that allow for the water to pass through very quickly through the membrane. In addition to water, you also reabsorb many of the nutrients that your body needs. So glucose, amino acids, vitamins. These are also being transported out of the filtrate back into the bloodstream. These nutrients are very important for the body, so you don't want to just waste them. Remember, if it stays in the filtrate, it's going to be concentrated and it's going to leave the body through the urine. And so you don't normally have glucose in the urine. You don't normally have amino acids and vitamins and things in the urine unless you're overloaded. Now that can happen. You can have, because each of these has a carrier type molecule, so it's facilitated type diffusion. Each of these has a carrier type molecule that can be overloaded. So for example, if you have a very high glucose diet, or if you're diabetic, then yeah, you're going to find glucose. You can detect glucose in the urine. That's because there's more glucose more glucose molecules to be transported than you have actual transport proteins to get it across. And so so there is a limit there. The number of carrier molecules is what's limiting the transport and creating a transport maximum for these types of molecules: glucose, amino acids, vitamins, any of these nutrients. Moving down to the loop of Henle, the next set of tubes, there is some filtrate that gets, the filtrate does get reduced here, estimated by about another 20%. Some of that, there's different types of cells in the loops of Henle. So the descending loop is mostly simple squamous epithelial cells, which happens to be very permeable to water. So most of this filtrate that gets reduced here by the 20% is taking place, is reabsorbing here in that descending loop. The ascending loop isn't as permeable to water, and so, but it is permeable to like sodium, and so some of the sodium re-enters the bloodstream based on the ascending portion of the loop of Henle. The distal convoluted tubule and the collecting ducts, these are also areas that some filtrate can be reabsorbed, but most of the filtrate is being reabsorbed in the proximal tube and the loops of Henle. Now the distal convoluted tubules and collecting ducts are being regulated through hormones like ADH and aldosterone, and so we've mentioned both of these hormones really throughout the course. ADH, antidiuretic, is working because it causes these tubules to become more permeable to water. So whenever ADH is secreted and it makes its way here, these tubules become more permeable to water, so that means that more water comes out of the tubules, which comes out of the filtrate and goes back into circulation. And so that actually helps in the reabsorption process. That's why ADH is retaining fluids, is because it's causing the filtrate to be, more filtrate to be reabsorbed. Aldosterone is kind of doing the same thing, but instead of reabsorbing filtrate directly, it's reabsorbing the sodium. Now, step 3 in excretion is tubular secretion. I just want to mention a few things about this. First of all, the definition is here: substances not normally produced by the body are secreted into the filtrate from some of the capillaries that are surrounding the tubes. Those are called peritubular capillaries. And so I'm bringing you back to this little figure diagram. And now we can kind of go through the process. You know, this is where filtration is taking place inside the capsule, inside the renal corpuscle. That's what the F is, that's filtration. Then you're making all the filtrate, and then everywhere you see the R, that's your reabsorption. So the proximal tubule does a lot of reabsorption. The distal tubule, the descending loop, mainly the loop of Henle, mainly the descending loop, not the ascending loop as much. And then you have the distal tubule here, and even the collecting duct does a little bit of reabsorption. The last stage that we haven't talked about, the S, that's called tubular secretion. In this case, you are secreting waste products directly from these capillaries into the tubes. All right, so make sure you get the direction correct. The tubular secretion is going the opposite direction of reabsorption. So it's kind of the opposite of reabsorption. You're secreting substances, waste products from the capillaries directly into the tubes. Usually it's because that these things, such as hydrogen ions or other drugs like penicillin or maybe some metabolic waste products, they didn't get through the filter here for whatever reason. Maybe they were too large to go through that filtration membrane. Remember we talked about the different layers of the filtration membrane. Maybe these were too large. Maybe they didn't have the right ion charge associated with it, and they were repelled, and so for some reason they didn't get out of the filtration membrane. Well, now they have another opportunity here to actually diffuse out and be transported out of the capillaries into the filtrate, and that's called tubular secretion. The last couple of things I want to mention for this chapter is urine movement. Now that we've created the urine, we've created the filtrate, we concentrated it down, we regulated GFR and all that, and so now we've created the urine. Just a couple of things about urine movement. Hydrostatic pressure, that's water pressure, is basically the driving force to push the urine through all the different nephron tubules. It's just water pressure that's there. That's how the urine and the filtrate is moving through the nephron itself. Once the urine is produced and it goes through and leaves the kidneys, it's going to go into the ureters. Well, these are muscular tubes that help in the process of peristalsis. They help in the process of urine movement through the process of peristalsis. Peristalsis is the same type of contraction movement that we talked about when we looked at the esophagus and the digestive tract. The smooth muscle contractions, wave-like motions that are pushing something through the tube, and that's what's happening for the ureters. They're pushing the urine through these muscular tubes. Every few seconds, every couple of minutes, they're contracting, and that's pushing the urine through the tubes until it reaches the bladder. And the urinary bladder, is what's going to store the urine until it is released through the urethra. Which then brings us to what's called the micturition reflex. This is urination. This is a reflex action. The diagram kind of goes over this. Figure 24.27 is going to go over some of this. You can kind of see some of the steps here. They don't show you the ureters coming in, but they would kind of coming in down here to the sides, and the urine is going to fill up the bladder. It's going to cause the bladder to stretch. Remember, the urinary bladder is going to be made out of smooth muscle as well. So again, just like any other smooth muscle, once it stretches, it'll create a contraction reflex. And so once it's starting to stretch receptors send the signal through the nervous system, afferent neurons, and it goes over to the spinal cord. So it's sending a signal to the spinal cord, and you have a reflex type action. That reflex is sending a signal back to the bladder to help you start releasing and opening up or relaxing the sphincter muscle. So here's the start of the urethra. Notice you have two sphincter muscles. These are bands of smooth muscle that are kind of creating like a rubber band effect around the urethra. There's two of these. You have an internal and external. This reflex action is reflexing back, and it's basically telling this internal urethral sphincter to start relaxing, start opening up, so that the urine here in the bladder can start leaving, to start being excreted. But you have to be able to control this, right? So there's actually, your brain has to send a signal, a direct signal from the brain through the spinal cord to the external urethral sphincter to get it to relax. And so this is the voluntary control of the diagram. This is the involuntary over here. Involuntarily, it's trying to, whenever the bladder fills up, it's trying to make you, go urinate. But you're able to control that. You have to voluntarily control, send a signal from the brain through the spinal cord to release, the pressure on this external sphincter to get it to relax, and that allows for the bladder to be voided and to urinate.